You're listening to Medscape's In Discussion series on endometrial cancer, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Ursula Matulonis, Chief of the Division of Gynecologic Oncology at the Dana-Farber Cancer Institute and Professor of Medicine at Harvard Medical School. She is the first recipient of the Brock Wilson Family Chair at the Dana-Farber Cancer Institute. She co-leads the Gynecologic Cancer Program within the Dana-Farber Harvard Cancer Center, the Ovarian Cancer Specialized Program in Research Excellence, SPORE grant from the National Cancer Institute within the Dana-Farber Harvard Cancer Center, and an NCI P01 Program Project grant that is focused on targeting replication stress in high-grade endometrial cancers. Relevant disclosures can be found with the episode show notes on Medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, I'm Dr. Ursula Matulonis. Welcome to Season 2 of the Medscape In Discussion Endometrial Cancer podcast series. Today, we'll discuss molecular classification and the genetics of endometrial cancer. First, I want to introduce my guest, Dr. Katherine Fuh. Dr. Fuh is an associate professor of obstetrics and gynecology at the University of California, San Francisco. She serves as the director of basic and translational research. She also serves as the John A. Kerner Chair in the Division of Gynecologic Oncology. Dr. Fuh was also recently appointed as the Community Engagement Liaison to the Molecular Oncology Program and the Office of Community Engagement at the Helen Diller Family Comprehensive Cancer Center. Dr. Fuh is a board-certified gynecologic oncologist and is a leading expert in the management and treatment of gynecologic cancers. She also serves on national committees for clinical trials and translational research, including serving as the translational co-chair for the Uterine Corpus Committee for the NRG Clinical Trials Group. And Katherine is the principal investigator on multiple grants from various federal and foundation sources, and she also runs her own lab. It's really an honor and a privilege to have Dr. Fuh here today, really a leading expert in so many aspects of gynecologic oncology. So Dr. Fuh, welcome to the Medscape In Discussion Endometrial Cancer podcast.
Thank you, Dr. Matulonis. It's very kind of you.
Well, you're fabulous, and it's great to have you here. So we're going to talk about a lot of different topics today, including you and your incredible career so far and where you see your career going. But just can you start off by telling us how you think about the molecular classification of endometrial cancer and how it informs treatment decisions for your patients?
Yes, no, this is definitely a topic that we're all thinking about and talking about right now. And I think about molecular classification actually defined as the original TCGA and PROMIS molecular classifiers, but also by immunohistochemical staining, such as HER2, ER, and PR. And I think that the way the TCGA was initially published in 2013 was really important. It laid that foundation for thinking about endometrial cancers more as molecular subtypes rather than histologic subtypes. And so, in this category, really thought about POLI, which are these ultra-mutated mutations in the exonucleus domain of POLI gene. And the TCGA found that patients with tumors with POLI mutations had the highest survival. And this really changed the way we approached and think about this. And there's clinical trials that I'm sure we'll talk about later that really are trying to understand how to use this information to become more clinically relevant. The second subtype is microsatellite instability high, which these are also hypermutated. And the two remaining subtypes are copy number low, which actually correlated more with endometrioid histology, and copy number high, which are more serous-like. And in the copy number low subtype actually were found to have increased progesterone receptor expression and, suggesting responsive to hormonal therapy. And again, this is something that we're really looking to as well in clinical trials. Copy number high cluster found to be more serous subtype, and actually might have more transcriptional activity of cell cycle deregulated genes like CCNE1 and PIK3CA MYC, as well as TP53. So it's really, helps us to inform clinical care, but also clinical trial development, which I'm really trying to move this into standard of care. The PROMIS molecular classifier, as we think about it in terms of POLI, mismatch repair deficiency, other subtypes, we think about it based on p53 immunohistochemistry and separate that into the NSMP or no specific molecular profile, as well as p53 aberrant or mutant. And this again is a way that we can think about, how do we translate to our patients? How do we use this to then think about chemotherapy plus immunotherapy, as we know from the 4 pivotal trials that have really helped us counsel patients for including immunotherapy with mismatch repair deficiency or MSI high tumors.
We've had discussions from Ginny Fleming about the role of checkpoint inhibition in mismatch repair proficient tumors. Panos Konstantinopoulos spoke about targeting the estrogen receptor for the non-specific molecular profile tumors. So we've had a lot of discussions about therapies, and certainly as we move through this podcast, would love to hear what you think is exciting in endometrial cancer. I think there's a lot, but kind of what your thoughts are. So really focusing on the clinical aspects of genetic testing, how do you see ordering genetic testing? I think we all see patients who come in who you can't find the sequencing, you can't find HER2 results. How do you order these different results? How do you organize them in the chart? And when do you order the molecular testing?
Great questions, because that really informs clinical care, and our patients are coming to us saying, gosh, what is molecular testing driven care? As we're thinking about this molecular testing, I think about in 3 broad categories, actually. So there's the ones that we're kind of used to having in-house for most, I would say, providers in terms of pathology performs, the immunohistochemistry for mismatch repair proteins. I remember as a fellow, that was something that was just getting started and really being launched. And now it's universal. Usually, I would say for most. So I think that's one way to think about molecular testing is really immunohistochemistry. And with mismatch repair protein, IHC, also estrogen receptor, progesterone receptor, and then in some places also HER2, IHC. So that is one category. The other molecular testing I think we talk about a lot is also next generation sequencing, DNA and RNA sequencing, using that, whether or not it's in-house, but for most it's commercially based, right? So we send that out. And the third category is whether or not it's all inclusive, both the protein as well as the NGS together, the DNA and RNA. Well, then that's done in-house or commercially as well. So I think when someone comes to us either as a new diagnosis, just having had surgery and has, let's say, advanced stage disease, so now what's next? Do we do the comprehensive or we do the DNA, RNA, and protein? I do for advanced stage. I think that's something where we can really have biomarker informed care, especially with if someone's mismatch repair deficient, we know combining with immunotherapy is critical. And then let's say someone who comes in with recurrent disease, they were treated somewhere else, or they were, initially early stage and now recurrent. Yes, I think that's another category because we know the clinical trials also correlate with improved response with knowing the molecular profile, especially again with mismatch repair deficiency or MSI high. Or then with the other biomarkers, HER2, for example, we know with PanDestiny trials that, that also correlates with response to TDXD and ERPR2, right? That also can inform. And also, what can help patients get into clinical trials? So this also helps with not just standard of care, but what are the other options for that? I think with early stage, there's some trials that we're still waiting for reading out. GY020 that looked at high intermediate risk early stage cancers with mismatch repair deficiency. If you combine with pembrolizumab with radiation therapy, is that better than radiation alone? So I think for the early stage cancers, we're still waiting. And then in terms of, how do we use POLE to inform us? And that's really more in the clinical trial right now. So I think in terms of the here and now, what we can do in terms of sequencing, how can inform clinical care, the advanced stage recurrent cancer patients, certainly having that molecular testing, both DNA, RNA, but also protein, I think is really important. And then certainly more to come for the other subtypes and stages.
What do you do about high risk stage 1 patients, like a stage 1 carcinosarcoma or a stage 1B? I'm using the old classification now, but basically muscle invasion serous carcinoma. Do you get testing on those patients, or do you sort of say, okay, you know what, let's give them a break and hopefully they'll do okay before ordering if they do recur?
I counsel, I talk about it with the patient, but I do tend to want to test. I do tend to want to have more information. I think it's important to have information to be able to help guide. And I think especially with the histologic subtypes we know can be more aggressive because they also have molecular aggressiveness in terms of the carcinosarcomas and the serous cancers that have more p53-driven tumors that can help us, guide HER2 expressing. We know that p53 mutated tumors tend to also have HER2 expression, whether or not that can also help inform the sequencing of treatment options. So for those, I would say like stage 1A, grade 1 endometrioid histology, endometrial cancer, I think we have sufficient data that those tend to do quite well with surgery, but certainly keeping our ears and eyes open for surveillance and making sure. But yes, I would say the more aggressive subtypes are the ones that I would also lean towards molecular testing.
Do you order germline testing on any patients for looking for BRCA1 or 2 or other?
Well, I would say Lynch is sort of where we think about it more, especially in the young patients, 40 and younger of endometrial cancer, even if they have risk factors that can, lead to endometrial cancer, we still test for hereditary and germline. For BRCA, we know that certainly, it is possible. So certainly with family history. But I think the way we do our panel testing, as you probably do in other people, it's a panel test. So if we're going to test for Lynch, we're going to also test for BRCA and the other possible genetic germline genes. So I think, yes, that's also that we consider.
Do you have a special place where you store all this information? It's, I was just seeing a patient just yesterday, and in the chart from the outside notes, the tumor was described as HER2 negative, which always drives me crazy without kind of a quantification around it. And then lack of source data for the sequencing. How do you store that information so it's easy to find for you and your colleagues?
Yes, exactly. That is a universal question. And I'm glad to hear it's a universal issue as well, too. I tend to store it in my progress notes, but I agree that until it gets to that progress note, it is a lot of trying to put everything together, which that needs to be improved for our patients and for patient care and for care as they're, maybe seeking other opinions and trying to understand. So I also agree with you that in terms of using our language better. So instead of saying HER2 low or negative, like what is actually that percentage and who did the testing too? And also, was it breast or gastric? If we want to get into the details that may help us inform in the future. But for now, and I think as an institution, we are trying to figure out how can we better store this information so it's more accessible for everyone, especially as folks are going through clinical trial and trying to understand, what are the options based on trying to find the information quickly.
You wrote a really nice commentary in Gynecologic Oncology in 2024 about whether we should be rebiopsying our patients with endometrial cancer at the time of progression to see if the molecular profiling has changed. And can you talk about what you recommend?
Yes, absolutely. This was a wonderful commentary that Dr. Beryl Manning Geist, who's now at Emory, and I wrote together. And it was from a case report from Britta Weigelt's group, written by Soshmita Gord-Handes, who really looked at 2 recurrent endometrial cancer cases and tried to answer the question that we ask quite often. Someone presents with either early stage or advanced stage, and this case report actually showed 2 cases. One was early endometrioid, one was an advanced stage 4B grade 3 endometrioid. Both had standard of care treatment and both had recurrences, one at 38 months and one at 6 months. And they were able to biopsy and test the primary tumor as well as the recurrent metastatic tumor, and actually found that along with sort of their typical poly wild type or mismatch repair proficient, they also found like a subclonal beta-catenin mutation or CTNNB1 mutation in the primary subclonal. So subclonal meaning that, probably doesn't do much. It's just sort of sitting there. But then in the recurrent metastatic tumor, they actually found that it became clonal. The recurrences were in the lung for the one with early stage, and for the advanced stage was in the brain. So certainly areas that can be difficult to treat as well too. And so I think this really highlights this idea that there is evolution, and these subclonal mutations can become clonal. And I think we're now at the stage of, well, now how do we actually target and actually treat these clonal mutations? And that's where, we sort of in the process of identifying these specific biomarkers that may drive these tumors to become more aggressive and more metastatic and recurrent. But now let's find the therapy that we can to pair with this and actually treat this. So I think this was a really nice example of perhaps rebiopsying actually might really be informative.
Any sense from what you've done that there's any change in HER2 expression, ERPR expression? I know the breast cancer docs talk a lot about that. And then, obviously we're going to be using more antibody conjugates in endometrial cancer, and whether or not folate receptor alpha changes or any of those other markers change over time.
Exactly. I think we ask this and talk about this all the time. And I think we're waiting for that information, as we are being able to accrue either retrospectively or prospectively. I know there certainly are studies underway right now trying to understand, how true. So if you have HER2 that is a tumor that does express HER2 at the primary tumor, let's say 5 years, 8 years down the line, is it still going to be HER2 expressing, or is something going to happen? And what are the treatments in between actually to help guide that? So hopefully we can all work towards this goal to understand that, because I think it's really important for our patients. We are currently using baseline tumors, I think, for most of what we're doing, because that is what we have access to, both in terms of, I would say, coverage. Sometimes we can't cover that second biopsy. So therefore it's difficult to do that, or maybe it's in a unique location that's difficult to have. But I think having a large scale study where we can answer that question would be really useful for our patients.
I want to move on to some of your research. Congratulations on the Route 66 endometrial spore grant, a specialized program of research excellent grant that you share with the University of New Mexico and University of Oklahoma. And you were involved in project 2 of that grant, which is entitled Inhibiting AXL to Improve Treatment Response in Endometrial Cancer. I know, and I've heard you talk on a phase 3 study that you ran of the AXL inhibitor AVB500 combined with weekly paclitaxel versus weekly paclitaxel alone in platinum-resistant ovarian cancer. And you also published a really fantastic paper in Cancers a few years ago that tested preclinically whether inhibiting the receptor tyrosine kinase AXL with AVB500 combined with bevacizumab would improve the response in uterine serous cancer. Can you tell us about AXL, what happens when it's inhibited, the results of this phase 3 trial on ovary cancer, and how those clinical trial results may have informed the clinical trial in the endometrial spore grant?
Thank you so much. And your group has really led the way with these multiple spore grants as well, which are really important in GYN cancers, really gives us an opportunity to expand from the bench to the bedside. So yes, this project with the AXL inhibition to improve response in endometrial cancer started when I was at WashU, and now carrying forward now at UCSF. And the way we developed this spore endometrial cancer project was from our preclinical data. And that preclinical data showed that when we inhibit AXL in endometrial cancers, we can improve sensitivity to paclitaxel. And we have some mechanisms to show that. Michael DeBoni, who's now at University of Alabama, worked on inhibiting AXL and combining with bevacizumab, and showed in uterine serous cancers, it really also improved response and decreased tumor burden in preclinical models. In parallel to all this, I was leading the phase 3 clinical trial, as you mentioned, in platinum-resistant ovarian cancer. And this was a really important trial because we looked at, standard of care, weekly paclitaxel versus standard of care paclitaxel with the AVB500, which now we call Batiracept, in platinum-resistant ovarian cancer. And at the time, similar to uterine cancer, same with ovarian cancer, thinking about the AXL protein by IHC as a biomarker, we had found that a majority of the tumors express this biomarker. So we included everyone in this platinum-resistant ovarian cancer trial. And what was informative was that when we looked at AXL high tumors, those patients actually really responded well and had improved both progression-free survival and overall survival in ovarian cancer when they received the paclitaxel with the Batiracept or the AXL inhibitor in ovarian cancer. So this was happening at the same time that we had the spore open and developed this uterine serous cancer trial. So this trial is actually going to move forward soon. Hopefully we'll open at 4 sites, Washington University, University of California, San Francisco, University of New Mexico, and University of Oklahoma, and really give our patients opportunity to receive an AXL inhibitor with paclitaxel. And actually, because of the ovarian cancer trial, we're now working with Exelixis for their AXL inhibitor, zanzalintinib, just based on the timing of everything. So in general, we're really excited to see, how we can actually bring a sort of a new pathway or inhibit a new pathway in GYN cancers to give additional options for our patients.
Yeah, that's great. I remember hearing you talk about this. The trial has started?
Yeah, it's going to start. We're in the final phases of the IND at the FDA. So that was submitted early February. So we're hoping if everything goes as usually scheduled and planned, that we would be able to hopefully open in the spring. So we'll, of course, see how that goes.
Good luck. That's great. Great work, and really exciting to hear about the project. And certainly endometrial serous cancer is, we still need really great therapies for this histology. I also want you to talk about, because you're involved in so much at UCSF and elsewhere as well, obviously the SPORE and the NCI, but can you talk about other novel and noteworthy projects that you are working on at UCSF right now?
Oh, thank you so much. We're really looking at the tumor microenvironment and trying to understand how fibroblasts, usually thought to be very supportive cells, can actually, with expression of either AXL or another receptor tyrosine kinase, DDR2, can actually influence tumor cells. And actually, by inhibiting AXL or DDR2 in the fibroblasts, we can actually lead tumor cells to be more responsive to treatments, such as PARP inhibitors. We have some data that's going to be hopefully published soon where we can actually show and highlight that by manipulating the fibroblasts, we can actually regulate tumor cells to become more sensitive to PARP inhibitors because they are suppressed by these fibroblasts expressed DDR2 or AXL. So that's one angle that we're looking at. Another is working with a collaborator at the Innovative Genomic Institute at Berkeley and Alan Ashworth, where we're trying to deliver cytokines specifically to the tumors through lipid nanoparticles. And we have some data already in our preclinical models where we can actually focus this on the tumors themselves and actually looking at the tumor infiltrating lymphocytes, see more immune activating T cells in these tumors. And so, of course, we're now going to pair it with other immunotherapy and see if we can, again, offer additional options for patients who have tumors that typically are not responsive. You've led these big clinical trials too, looking at this already in ovarian cancer, but trying to really figure out a different angle and more tumor specific possibly.
Well, it's exciting. I also just want to talk about you. You have so many hats and roles. You're the director of translational research at UCSF in gynecologic oncology. You run trials, which you've just talked about. You have a lab. Importantly, you operate on patients as a gynecologic oncology surgeon. And I also imagine that you mentor junior faculty as well. So I'm just wondering, how do you do it all? How do you run your day? And what advice would you give to junior faculty fellows who precede you to be as successful as you are?
Oh, thank you. That's very kind of you. And I think I actually would say that it's, looking at role models like yourself and having excellent mentorship early on. I really do attribute that a lot with the time at WashU and the mentorship there, really giving the time to build and establish this foundation for the lab, especially the lab-based research. You really need that. And so I would say Greg Longmore, who's actually a medical oncologist, molecular oncologist, he was integral, Dave Mudge, Matt Powell, Pramod Thakur, they all really, helped to carve out that time to establish the lab base and then also a really wonderful supportive team. So we had a PhD, Deb Frank, who really, I wouldn't say guide the grant writing, but just really, RFAs that are coming out and just really feeling that team spirit, which is really important. I think for the young folks who are coming through the pipeline is really figuring out your motivation for all this. I would say for me, it's the same motivation that I had that had me into GYN oncology or these patients and seeing how we do need more therapies. And so I think that really helps to prioritize what is important in your day and how to get through everything. And I really do also want to highlight the clinical trial mentorship with Katie Moore specifically, who really helped guide this bench to bedside translation and all the time and effort. And also the GOG Foundation in terms of Tom Herzog and Rob Coleman and Dave O'Malley to really make this transition. It is not easy to go from the bench to the bedside, and it definitely takes a team of support. And again, role models like yourself and others have really also helped make me feel like I can do this because you've done this. And so I think that's, again, really important. So I appreciate all that support.
Well, that's very kind, but you are a star and you've accomplished so much and really look forward to seeing your career and your continued success. So I just want to finish up kind of on a clinical note from a therapeutic and clinical care standpoint. What recent research and findings in endometrial cancer do you think are important and noteworthy for our audience to know about, to learn about?
Yes, I think, antibody drug conjugates, we didn't really talk about that much during this time. And I'm sure you have other sessions that have addressed this with Katie Moore in the previous season. And I think that's something that it's a field where it is important. And we're really going to try to understand how can we sequence and utilize ADCs in endometrial cancer, whether or not they're really biomarker driven or are they just agnostic to the biomarkers and actually just thinking about, when and where to use it. I think also thinking about other ways for maintenance therapy. So, thinking about p53 wild type tumors and how do we actually have maintenance strategies? Are they estrogen receptor positive, progesterone receptor positive? Can we utilize the maintenance strategy based on that expression level or through selinexor with p53 wild type tumors? So I think that's noteworthy as well too, as we're thinking about how to deal with the tumor that's measurable in there, but then also how to maintain it, especially for those that are mismatch repair proficient, that don't have the immunotherapy as much as an option for maintenance.
Katie Moore spoke during the first series about antibody-drug conjugates, and coming up, having a discussion with Leith Ellison from Mass General He's a breast cancer researcher, and really what the breast cancer docs have learned about ADC after ADC, which we're really experiencing right now in gynecologic oncology, but really don't have great answers to. So what should we be thinking about? But I think you pointed out some really great research that's going on and very exciting findings. So thank you so much. Katherine, it's been great to have you on this podcast, and thank you so much for joining.
Great. Thank you so much. Appreciate it.
Today, we've talked to Dr. Fuh about molecular classification of endometrial cancer, new and exciting research that she's been working on, as well as others, and what's to come in the future, and also learning about how she's become so successful in her career, really holding on to multiple different hats in terms of administrative hats, research hats, mentoring hats, etc. So thank you all for tuning in. Please take a moment to download the Medscape app to listen and subscribe to this podcast series on endometrial cancer. This is Dr. Ursula Matulonis for the Medscape In Discussion Endometrial Cancer podcast.
Thanks for listening to Medscape's In Discussion Endometrial Cancer podcast series with our host, Dr. Ursula Matulonis. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out medscape.com or the Medscape app for show notes, links, and more information on endometrial cancer.